So our essential question, what are the different types of cells that make up living things? So not all cells are the same. Cells are the basic unit of life, but some are bigger, some are smaller, some are more complicated with more involved parts. Some are just very simple and do their job and go home. So our essential question is, I think it's the same for 3.1 and 3.2. Different types of cells that make up living things. The cell theory. We talked about theories way back in chapter 1. A theory is something that has evidence. So if something is called a theory, it means that thousands and thousands of scientists have performed experiments and found that these three things always, always, always apply to cells in this case. So all organisms are made of cells. I said to be an organism, to be a living thing, you have to be made of at least one cell. There are some things that are only made of one cell, and that's okay. They're still considered living things. All existing cells are produced by other living cells. Cells don't spontaneously generate out of nowhere. Once upon a time they did, and that's how we got living things in the first place, but that's a discussion and a lesson for a future chapter. And the cell is the most basic unit of life. And your guys' Ed puzzle this week is about how the cell theory came about and was formed, and what scientists had a hand in forming it. So all cells, no matter what, big cells, small cells, complicated cells, simple cells, all cells have these three things in common. And this slide is going to be important for your end of notes assignment, but we'll come back to that. So all cells have genetic material. We talked in chapter 2 about nucleic acids. Those are our genetic material. DNA and RNA are the main ones, but all cells have some form of genetic material, some nucleic acid they are made of. They are all surrounded by a membrane. A membrane separates the inside from the outside, just like our skin. Our skin separates our insides, keeps our muscles and our blood and stuff in. Good morning, Alex. And then keeps bad stuff out. So all cells have their own form of skin. It is called the cell membrane or the plasma membrane. You will see it and you will hear it both ways. They mean the same thing. All cells are filled with cytoplasm. There's that prefix again. Cytoplasm is the jelly-like material that just holds everything in place. I don't know if you've ever eaten a Jell-O salad, where you make the Jell-O salad and then you pour the fruit into it and the fruit is just kind of stuck in the Jell-O. That's what our cytoplasm is. It's the Jell-O that holds all of our little structures in place. It's semi-Jell-O. It's fluid, but not as fluid as water. It's a little thicker than that. There are two main types of cells that we're going to talk about today. The first type is called eukaryotic. And we do a lot of prefixes in science. The prefix "eu" as in EU means true. And the root word karyo means nucleus, means kernel. It looked like a kernel under a microscope, but we use it for the word nucleus. So these cells have a true nucleus. So in addition to a membrane and cytoplasm, these cells have a nucleus, which acts as the brain of the cell. We'll go into more detail about the nucleus next week. But it also has organelles. These are little mini organs that each have their own specific function inside of the cell. So just like we have our organs, we have our heart, our stomach, our lungs, our liver. They each have their own function inside of us as a whole. Our individual cells have their own little mini organs called organelles, and they each have their own specific function within the cell. So we will go through each and every one of those next week. Just know that eukaryotic cells have a nucleus and also have organelles. Have all the organelles. The second type of cell is a prokaryotic cell. So the prefix pro actually means before, and then karyo still means nucleus. So these cells evolved before the nucleus evolved. So these are ancient types of cells, and they're simple. They have genetic material in the form of DNA or RNA, but they don't have a true nucleus. Its DNA is just kind of floating in the middle of the cell somewhere. Doesn't have a lot of organelles, has a membrane, it has cytoplasm. It does have one organelle called a ribosome. Ribosomes make proteins. Proteins are what we need to live. So while it doesn't have all of them, like a prokaryotic cell, it is very simple. It just performs basic survival life functions. And this week, you're going to look at prokaryotic cells in the form of bacteria under your virtual microscope. Bacteria are considered prokaryotic cells because their DNA just floats around in the middle. It has that membrane. Some of them have a cell wall. Has the cytoplasm in the background holding everything together. All right. And that's actually it for the notes. 3.1 is very, very simple. So I'm going to go over the assignments. So your summary assignment this week, you're not writing a traditional summary. You guys are not writing the paragraph. You are drawing a Venn diagram. If you don't know what a Venn diagram is, it looks like that. So it's two circles. They intersect each other in the middle. Draw them big because you're going to write words in the middle. And you're going to list all the similarities and differences between prokaryotic and eukaryotic cells. You can list the ones we just went through in our notes. We have one slide. Like I said, it says all cells have all this stuff. That's all the stuff that goes in the middle. And then include examples of each in the different part of the diagram. So on one side, we'll have the prokaryotic cells. The other side, we'll have the eukaryotic cells. In the middle, you tell me what's the same. And then on the edges, you tell me what's different about each. Like, it doesn't have to be overly complicated. 2 to 3 bullet points each is fine. I gave you 3 for the middle portion. One of the bullet points on each side should be examples of each. So that's what we're doing for the summary portion of the notes this week. And then the other thing I want to show you is your microscope lab. So right here, today we're doing section 3.1 notes. You also have a virtual microscope lab where you are learning about the microscope. You're learning about the parts, all that good stuff. So it's structured as a quiz, but it's just an assignment. You guys can do it as many times as you want to get full credit. And the website you're going to be looking at looks like this. The link is in the quiz. It takes a minute to load. So I reloaded it. Sorry, looks like this. It takes a minute to load. You have to give it a minute. And then you have the microscope, and it'll tell you. Go through all of these. First one you're going to go through is learn. You're going to learn about all these parts of the microscope. The body, the eyepiece lenses, the diaphragm, the lenses, the nose piece, all that good stuff. And then on your quiz, there's also a microscope where you're going to match the parts. That's how I know that you actually went through it and matched the parts. And then back here, when you do all that, you're going to go to main. And then you're going to click on explore. In this box right here, that's where all of your microscope slides are. So you're going to click on the box. And the first thing we always do when we look at microscopes, you have to learn how to use the microscope. So all we do is look at the letter E under a microscope. So you're going to go on sample slides. Again, this is all spelled out in your assignment. The letter E, and then this comes up. So if you're actually looking under the microscope like this, it's a little bit blurry. So you have to focus it. And that's what these on the bottom, this coarse focus, you got fine focus, and you have light adjustment. Do you want more light? Do you want less light? You slide these back and forth until it comes into view. So coarse focus does it in big steps, and then fine focus does it in little steps. You can still see the difference, but it's little steps. And then light, really bright light doesn't always work. So light you usually want in the middle. And then you're going to draw that. You're going to draw. So one blank piece of paper. There it is. So you're going to draw a circle, and then you're going to draw an upside down E, because that's all that is. And you're going to label it 4x. So that upside down E is 4 times the size as it would be if you're just looking at it with the naked eye. All right. Then you're going to go to 10x. It's going to load again. It gets a little bit bigger. So you're going to draw another circle under 10x, and you're going to draw just the part of the E that you can see. I don't draw very well, but that's just the part of the E that you can see. And as you get bigger and bigger, you're focusing in on one small part. Make it clear, should be clear. That's just one small part you can see, but it's 40 times the size. As you can see with the naked eye, you can see it, just that one part of the E fills up almost the entire circle. And then 100x. 100x, we always have to add immersion oil. So you click the little question mark by the immersion oil, and then it'll go. And you can adjust it so the entire screen is now black. So you're just going to draw a circle and then fill it with black. That's your 4th circle. So by the end, you should have 4 circles with the 4 different drawings of the 4 different magnifications. You're going to take a picture of your drawings and you're going to turn them in. That's how you do that. Then you remove the slide. When you have finished removing, it's very important to thoroughly clean it. So you get the lens paper. Oh, those are chem wipes. See, I did that wrong. That's not what you want, the lens paper, which is this one. Then you clean it off, and then it'll reset. And then you go again, and it'll tell you you got to do your second slide. Those will be the bacteria slides. It's the same process. It's the same questions. And then next week, you'll be looking at even different cells from this exact same website. All right. Does anybody have any questions about the assignments this week? All right. Some of you still need to take the test. Some of you still need to take the Chapter 2 exam. Michaela, I hope that was for no questions and not the exam. So if you have not taken the exam, Go do that now. It's going to close all your tabs. You have to close all your tabs to be able to take the exam.